how to determine the order in which the different nucleotide bases of the DNA strand are present. So during the several decades, past decades, now this technology, sequencing technology has increased exponentially. And so now we have the very good ability to sequence the genome, and almost there are several clinical labs also all over the world which have come up, which can provide accessible and cheaper economic ways of sequencing the DNA. Now, the first major concept or foray into the sequencing came up with the advent of the Human Genome Project, which is almost a $3 billion project, and it took almost 13 years, completed 99.9% completed in 2003. So this took almost 13 years for sequencing the entire genome. Now this Human Genome Project was accomplished with the so-called, what is now called as first generation sequencing technologies, which is also called as the Sanger sequencing. So this Sanger sequencing, which is also called as the chain termination method, it was developed in 75 by Edward Sanger, and it is still considered the gold standard for nucleic acid sequencing. For the subsequent, actually next 2.5 decades, this was the only technology that was available. If you look at the various milestones of the sequencing development throughout the history of molecular biology, way back in 1975-77, two important technologies came up, which is now we call them as first generation technologies. We call the Sanger sequencing and then the Maxim and Gilbert sequencing. Then the after 2000 onwards, what we call as next generation technologies came up. And in 2005, we have what is called the pyrosequencing technology. 2006 saw the advent of Illumina. 7 saw the solid system, and around 10 we got the ion thorium. You know, these are all considered the second generation technologies. Then after 2010 onwards, there is a development of newer technology called the third generation technology or next technology or TGS, third generation. So here we have single molecule sequencing from PacBio, SMRT sequence, this what's called, and then you have the nanopore sequencing. So broadly, we can classify them as first generation, second generation, and third generation. So we'll briefly look at these technologies. So first generation technologies, mainly Sanger and Maxim, and they infer the nucleotide identity using BNTPs, and they separate them with electrophoresis. The average size of the fragments was ranging from 500 to 1000 base pairs. The second generation sequencing, like the ion, torrent, Illumina, these are called high throughput or massive parallel sequencing. So multiple DNA pieces have been sequenced in parallel multiple times, but the read lengths are only shorter, 50 to 500. So these two together, they are called short read sequencing technologies. The next one, which came with for third generation, what we call, or single molecule sequencing, these developed by PacBio and Oxford, they sequence the native DNA in real time. There is no amplification. The first two technology directly from the native DNA in real time in single molecule resolution we can sequence. These are called long read sequencing because tens of kilobase length of the fragments can be sequenced on average. So briefly, the first methods, the earliest methods were the DNA sequencing, that is the chain termination method by Sanger and the chemical degradation method by Maxim and Gilbert. Now, to start with, both were very popular, and most of the labs during those times, they were doing either the Sanger chain termination or chemical degradation method. So both were quite popular, quite laborious at that time when they developed it. And the basic principle is that single-stranded DNA molecules, which vary in length by just one nucleotide, can be separated using a polyacrylamide gel. So there is long sequencing gels which are, so it is a very laborious process at that time. And this is the chemical degradation method, which uses specific chemicals to degrade the DNA after specific sequences. After that, so there will be 4 reactions. Then it has to be run on and they should be radio labeled. It is run on a sequencing gel, very thin, ultra thin polyacrylamide gels. Then you subject it to autoradiography. So it's a very laborious process. The dideoxy method or the chain termination method, it uses a unique nucleotide, is called the dideoxy. There is no 3' OH group in the DNA. So in the nucleotide, there is a normal 2' deoxy. This is dideoxy. Both 3' and 2' lack OH group. Because of that, it leads to chain termination. So one of the four is radio labeled, and this is incorporated into the growing chain by polymerase, and it will stop at that specific length. Now, this chain termination method came up in a very big way because of the availability of the automation. So there is now this process was automated. Radio labels were replaced with fluorescent labels. Each DNA, each radioxy nucleotide was labeled with a different fluorescent label. So it gave different labels, fluorescent dyes to each of the DNA pieces, which were separated not on polyacrylamide gel but on capillary machines, capillary electrophoresis, which because of which it was completely automated. And now we have, this is still under use. So we get this what is the chromatogram. Each piece indicate a particular base, and it can be read. So the automated DNA sequencers like the GA3000 from Applied Biosystems and all, they came up. You have the single capillary machine, you have the 8 capillary systems, and then you have the 384, 96 well capillary system, and then 384 capillary system. All this high throughput type of systems came up for separation and then for analysis using fluorescence. So this is a typical large scale DNA sequencing facility somewhere in US, which can sequence around 120,000 DNA sequences from 60,000 plasmid depth. The original human genome sequence was sequenced using this kind of facility, large scale. So what was wrong with that? Now this method, dideoxy method, is good only for 500 to 700 base pair reaction. That's the normal read length, and it is quite expensive. It takes some time for sequencing, and the human genome is around about 3 million base pair. That's why it took almost over 12 to 13 years to get a complete human genome draft. So then the next generation sequencing came up because of the increased demand for cheaper and faster sequencing methods. So the main basic idea here is these perform massive parallel sequencing, that is large scale and parallelly multiple times, during which you have millions of fragments of DNA from a single sample. They are sequenced in unison. That is, at the same time, they are sequenced multiple times. So there is a very high throughput, and the entire genome can be sequenced theoretically in less than a day. So this is sequenced by synthesis. Advantage is very accurate. There is parallel processing, and it can be automated. There is no gel electrophoresis. So gel electrophoresis was the major drawback in the second generation technologies. Here there is no electrophoresis here. So several during the 2000, early 2000, several next generation sequencing platforms came up across the world. And one of the pioneers were the Roche system for 454 sequencing system. They came up with several machines like GS Junior, GS Flex. Then Illumina came up with several machines. Applied Biosystems came up with a solid system. Then the ion torrent is another revolutionary technology which is based on semiconductor detection based technology. Then Helico, up to here. So these are 1, 2, 3, 4. 4 are the important next generation sequencing technologies. But to start with, several of them came. But currently in the market, only 2 are running: the Illumina and the ion torrent. Only these 2 technologies could withstood the vagaries of time, and they're still running. Whereas the others, because of some of the other drawbacks, they're no longer in use now. Then the third generation sequencing came up. Helico started with that, but still they could not carry forward. Now we have this Pacific Biosciences or PacBio RS. They have the system, and then the Oxford nanopore technology, the nanopore sequencing and the PacBio sequencing are the third generation or next generation sequencing, TGS. So a typical second generation, we look at the second generation workflow first, then go to TGS called NGS. So they basically involve, you have to prepare a library. That is, you have to amplify your DNA sample. So you have the library preparation, which involves either emulsion PCR or what is called as Polony PCR on a slide, glass slide. So the semiconductor technology, sequencing technology of ion torrent uses emulsion PCR. Pyro sequencing also uses that. And then you have the solid, this also is emulsion PCR. Whereas the Polony PCR of the Illumina, the Illumina reversible terminator, it uses the Polony PCR for library preparation and amplification. So there is a library preparation, amplification, then sequencing, 3-step workflow. So library preparation generally involves, we just take a small amount of DNA. All this NGS sequencing requires very small amount of DNA, around less than microgram. So maybe around 200 to 300 nanogram of DNA theoretically is sufficient. So DNA is collected and then it is fragmented, random digestion by sonication or enzymatic. Then we have what are called adapters. Adapters are small DNA sequences, pieces of known sequences and the barcodes. They are attached, ligated to these pieces. They are all very random. On either side, on one side you have an adapter, another side you have got a primer. This gets attached to the DNA, and this is how you get. You select the pieces which contain both these, and then go to the next couplet. So next you go to the emulsion PCR. So library preparation is common for both. So in emulsion PCR, what happens? As the name indicates, you are carrying out the PCR in an emulsion. So there's an emulsification reaction which contains an emulsion oil, all the PCR mixture, typical DNTP, then all the other components. You have the called micro beads. These are basically micro reactors, and then you have the library DNA which you have generated. So you mix it and make an emulsion. When you have this emulsion, you have the small micelles which are formed, and each one will be like a micro reactor. A typical bead should have, an ideal one should have this many components. There should be only one strand of the library DNA. There should be primers, and there should be only one bead. Now these beads will contain the small primers which are complementary to the adapters. So you start the PCR, typical three-step reaction. During denaturation, the library fragment here get denatured into two strands, and during annealing, out of these two strands, one of the strand will anneal to the microreactor bead. Now, microreactor bead has got small primers attached to that, and that adapter sequence what is attached is complementary to that, so it anneals. And in the third step extension, the polymerase which it will extend that to form a double-stranded. A single-stranded fragment is converted to a double-stranded. Then again you denature. Next step, what happens? The original the old strand which was attached here annealed, it comes off, whereas the newly synthesized strand remains attached to the bead. And this again continues every time new fragments are attached, and then it is extended. When you repeat it for 30 to 60 cycles, in the end you will have a bead, a microreactor which has got a bead with all the DNA pieces, double-stranded DNA pieces, which is amplified in a reactor. So this is the amplification step in the emulsion PCR. In the colony PCR, here this is called bridge amplification. This is the technology developed by Illumina, where they carry out the PCR on a slide, glass slide. It's called colony. So you have the colony of the fragments here. So here we prepare the genomic DNA sample, and then you ligate with the adapter, just like we did in library preparation. Then add this to the flow cell. This is a typical flow cell. In the flow cell, you have the glass slide, especially, and to which you have lawn of the primers which are attached. And these DNA are converted to single-stranded, and then they are attached, bind to the randomly inside the surface of the flow cell channels. Now this is how typically it goes. There are dense lawn of primers, and then you have the DNA fragment like this. It will be sitting there. So you start amplification, bridge amplification. First you add the unlabeled nucleotides and the enzyme to initiate. This is called solid phase bridge amplification. There is a bridge between the DNA and the primer here. So it binds, and then this is the primer, and this the adapter which are there, it will anneal to that, and this primer is extended. So it extends like a bridge. So it is extended like this. So the fragment, single-stranded fragment will become double-stranded. This will be happening throughout like that. And this is continued, denature, anneal, and synthesize, and then do the repeat cycle, you will get a bridge. So there is a clonal single molecular array of this fragment, single-stranded fragments, multiple amplified. So there are several million dense cluster of double-stranded DNA which are generated in each channel of the flow cells. So once you get the amplified the DNA, then you go for sequencing. Now the sequencing technology, I said there are four very important which came up, but currently we have only this ion torrent and Illumina. I'll just briefly tell the principle of this pyrosequencing, because this was one of the earliest developed DNA sequencing, next gen sequencing technology, by which is called the pyro sequencing, developed by the Roche. After emulsion PCR, they do pyro sequencing. Now, this pyro sequencing is a very simple, basic idea. The visible light is generated during the DNA incorporation by the polymerase, and this is proportional to the number of nucleotides incorporated in the chain. So if you have 1 picomole of DNA, this will be the number of ATPs, and it will give light. And what is basically measured is the light. Now, how the light is measured upon DNA nucleotide addition? There are 3 reactions, 3 enzymes are there. So you have the primer and the nucleotide. When you add, the polymerase will add the DNTP to the growing chain. Every DNA polymerase reaction releases a pyrophosphate. That's why it's called pyrosequencing. PPI, a pyrophosphate is released. Now, this pyrophosphate is made to react with another. See, the pyrophosphate is released. It is made to react with APS, adenosine pyrosulfate. So there is a sulfate there which is attached. So this is made to react with the APS, and the enzyme ATP sulfurylase will create an ATP and release the sulfate here. Now, this ATP is made to react with luciferin from with the help of enzyme luciferase from fireflies, and this is the one which is give out light. So for every nucleotide added, an amount of light is coming. So the light generated is directly proportional to the number of nucleotides added. So what is done? 3 steps. You immobilize the DNA, 3 enzymes are added, and then wash step to remove the nucleotides after each addition. So you have the polymerase which attaches, and the pyrophosphate is released, which is converted to ATP sulfurylase, which is converted to light by luciferase enzyme. So when there is incorporation, you get the light, and that is measured by a photomultiplier tube. So this is the flow cell, and they are the emulsion PCR. There are micro picotiter, not even micro, it's a picotiter plate. This is a typical plate. What you can see, this electron micrograph of that, the bead which is there, and this is the diameter of this 44 micron well, and this is the bead will be there, and the reactions will be happening there. So nucleotides, 4 nucleotides flow sequentially. You add one after another. You add ATP, like the DATP, see whether light is generated. If light is generated, that means ATP is incorporated. It takes it as A. Then go for C, then go for D, and then go for to like that. So after every addition, you check the production of light. This happens very fast. So when there is a light production, there is a peak. If there is no light production, then it will not generate peak and it will not be counted. So nucleotide addition order, you keep on going, and then it will measure the 3 reactions. So there is a photomultiplier which will measure. So that is the pyrosequencing technology. Somehow it was there for a couple of years later, and because of the competition and more alternative and cheaper technologies developed by Illumina and Anantar, and it was phased out.
ಅಲ್ಲ ಮಾರ್ಕ್ಸ್ ಸ್ಯಾಟಿಸ್ಫೈಡ್ ನೀನು ಬರ್ದಿರೋದು ಬಂದಿದೆ, ಇನ್ನು ಬರ್ಬೇಕಿತ್ತ ಏನು ತೋರ್ಸ್ ಏನಿಲ್ಲ. ಎಷ್ಟು 18 19?
I think somebody has to mute their mic. Okay, so we have the Illumina technology. There are several machines which came up, and this involves the library preparation, bridge amplification, and reversible termination sequencing technology. So these are the current Illumina platforms, Minisec, MySec, NextSec, HiSec, like that. And depending on the what size of DNA, how much of DNA you have to sequence. Like for example, the output for the simplest system is 7.5 GB. The highest one 6,000 GB output. That much of DNA. So read size, total read size is around 25 million. It can go up to 20 billion. So large amount of DNA can be sequenced. Parallel sequencing can be done by this Illumina platform. So this is a typical pipeline. You prepare the sample 1 to 5 days, ligate the adapter, do a bridge amplification for colony PCR. These are the flow cells there, and then you send it for sequencing, and then do the data analysis. So what happens? The sequencing chemistry here is what we call reversible terminator chemistry. So you add the sequence in the reagents, and the first base gets incorporated. Then you remove any unincorporated bases, you detect the signal. So for every base, there is a particular fluorescent tag attached for each of these nucleotides, 4 nucleotides, 4 colors. And on the 3' side, there is a block. Remember, the reaction happens, the 5' is attached, 3' will be free. So the 3' is blocked for every nucleotide. So that's called reversible terminator. It's a terminator sequence. It gets attached, but it cannot add next unless this is removed. That's called reversible. So there's a fluorescence dye attached to it. So this gets incorporated. So you do that first base, remove, then you incorporate it, you detect what color it has come, then you deblock, remove the block, and then remove the fluorescence and go to the next cycle. So this goes on. So this is a typical on the slide. This is what is happening for different molecules. So each incorporate all 4 nucleotides, each labeled with a different base. So wherever whatever is based on the template, it will add the color of the dye. And each piece of DNA which is hanging from the plate will add a different dye. And that is monitored by a camera. So basically, you are visualizing it under like a microscope. So each spot, different colors you can see. And that color keeps on changing after every reaction because this is added, next this is added, next this is added like that. So top, for example, this spot what is being shown, it is showing CATCGT, this spot upon sequence addition. The next one will be showing C like that. So depending on the color, each spot will indicate what base is incorporated. And this is done multiple times parallelly because this is on the plate, the camera will be there, and it is image analysis. You are basically analyzing the images, base calling from the images. For example, the top one, 1, 2, 3, 4, this is the sequence, and the bottom one at this region like that. So each spot you can call the bases, and these are all identified, put together, parallel sequencing, and then they're analyzed. So that's the Illumina sequencing technology. The semiconductor sequencing of the ion torrent is another ingenious sequencing technology. It's a very simple technology, which basically a glorified pH meter because you are measuring the change in pH, hydrogen ion concentration. So we have different machines, ion torrent and ion proton like that. And this is the ion torrent machine with, on an average 200 base pair. The length throughput is up to around 1 GB, and there can be 11 million bases. This is the older version. Now we have up to 60, 70 million bases per run can be carried out, and accuracy is almost 99. So these are especially semiconductor chips. These chips will be doing the DNA sequencing. So these thin wafers cut out from the silicon, and the transistor circuits are then pattern transferred, photolithography, and this is repeated. You are generating a circuit on the microchip. So there is a very simple natural chemistry and very rapid detection. So what happens? The microwells, these are the wells, okay, different layers, and this will be on the semiconductor chip, ion sensitive layer, below which is an ion sensitive field effect transistor, ISFEC, ion sensor. And each microwell will contain single standard template DNA molecule to be sequenced and one DNA polymerase. And these are the various different chips, different generation chips. This is 12 million now. Currently, we also have up to around 60 million sensors chip also. Yeah, this is the latest one where we have up to 60 to 80 million sensors. So that means that many number of molecules can be sequenced at a time, up to around 10 to 15 GB, and runtime is within around 2.5 hours. Analysis can take around another close to around 18 hours. So the chemistry is very simple. Now, upon addition of a DNPT to the template, it releases a hydrogen ion. Every reaction, polymerase reaction, releases a proton. It releases a pyrophosphate there, it releases a proton. So a hydrogen ion is released as a byproduct. Now, when there is a hydrogen ion, that is sensed. So basically, you are measuring a hydrogen ion, just like a pH. So this is basically a long, expensive, glorified pH meter. So bases are sequentially flowed into the cell one at a time. Upon incorporation, the nucleotide releases a hydrogen ion, which creates a pH change. And this chain is this will go and hydrogen ions will bind to the sensing layer below, which is a sensor plate, and that will transmit and it will create a voltage change between the source and the drain, and then it will measure. Okay, if there is an incorporation, it measures the change. So there is sequential addition of nucleotides, 4 nucleotides added sequentially. You add one nucleotide, see whether change in pH. So for example, T, there's a peak, G, there's a peak, like that. And if there is no hydrogen ion release, you will not get a peak. You'll get a, see, there's a gap here, and there are 2 T's, like that. So this is red. So this is corrected ionogram. This is how read from the sequences. So there are multiple, so how many bases are attached, T, like that. And then, so there are 4, the long sequence, so that means there are 4 T's. So basically, you are measuring the change in pH upon addition of individual bases, and then thereby recording the bases. So if you compare, these are the 2 prominent next generation sequencing platform currently in the market. Illumina and the Life Technology, which is now taken over by Thermo Fisher. Personalized genome machine, it's a small benchtop genome sequencing, and this can do up to 2 GBs. An SPI system can go up to 15 GB. Illumina, they have got a series of different machines and minisec, MiSec, like that. And these are various potential applications. You can do exome sequencing, targeted gene panels, and microbial exome sequencing, transcriptome sequencing, all those sequencing can be carried out by this. So that was about the second generation sequencing. Next, the third generation is called next. And this is also called as real-time sequencing of single DNA or RNA molecules. So here it is by synthesis, and nucleotide incorporation and detection happen simultaneously. As fast as a polymerase can incorporate nucleotide, on an average, 500 to 750 nucleotides per second is added, and that is measured almost at the same rate. And you have one system called SMRT, single molecule real-time sequencing, which is developed by Pacific Biosciences, called PacBio. Then without synthesis or ligation, it's called another technology called nanopore sequencing, developed by Oxford Nanopores. Now both these technologies give what we call a long read sequencing. The sequence which is come out will be up to 1 kilobases to 2 megabases, 1000 to 2000 sequences. But compared to the up to next generation, Illumina and Iontor, and they can give only up to 500 base reads. This will give long reads. Now, what is the advantage? Sequence assembly of short versus long reads. So these are the short reads. So the assembly becomes a problem in the short reads. The large amount of data, but when the long reads, it becomes much more easier to assemble. So long reads greatly simplify the assembly process, and it provides high quality alignments, and it will generate the finished sequence, which is sometimes a problem. Generally, when you are doing the entire genome sequencing of large organisms, 0.5 to around 1% of the sequence cannot be assembled properly. But this can be possible by long read sequencing. So what are these technologies? The SMRT, single molecule real time, developed by Pacific Bio. So in the reaction cell, they contain a single DNA polymerase. This DNA polymerase enzyme is immobilized on the bottom of a reaction cell, which is called ZMW, zero mode waveguide. This is a small cell, and there is a modified. The enzyme is a unique enzyme from a bacteriophage 529. It is modified, genetically engineered, and this is a fast single subunit enzyme. This is immobilized at the bottom. The enzyme is immobilized at the bottom of the tube, and each sequencing plate contains thousands of these individual cells, each holding a single DNA molecule. A DNA molecule is high complex. So the basic chemistry is you are adding a phospholinked DNTP, which uses a different fluorophore for each of the bases. Each one base, one color. So during the sequence, a single labeled DNTP enters the polymerase, and the DNTP is held in place shortly. And then the reaction happens, and the fluorescent signal emitted in the ZNW. In the bottom, you have the capturing, the emission happens, fluorescence. There's illumination, and there's capture of light, emission, and then it measures whether it is blue, green, red, or whatever color. And then the DNTP is released, it leaves, and then goes off. Now, this technology uses the fluorescent label linked to the phosphate. The earlier reversible terminal technology and other technology, earlier technology, they label to the base. This labels to the phosphate. Now, what is the importance here? Because during the chemical reaction by polymerase, these two phosphate, alpha and beta phosphate, goes out as pyrophosphate. Because of this, when this base is linked, then the fluorescence goes out. So when it is attached to the polymerase only, it is captured. Once it is released, it goes off. Okay, so the phosphate chain is cleaved from the incorporated DNA. So upon incorporation of a link, the DNA polymerase cleaves the dye and it leaves the phosphate chain. And this dye phosphate chain quickly diffuses to a short distance out of the detection volume, ensuring the background remains the same low level. So this is how it happens. You have the DNA polymerase, which is immobilized. Then the single-stranded DNA template is added, attached to that, and the DNTPs, each with different colors, they bind. Upon binding, now you see the bottom, the intensity of the light. It binds, the fluorescence is measured. There's A, this is green color, A is bound. Then you have it goes off, then you have the P bound there. And once it is attached, it releases the phosphate, it goes out, it cannot be detected. So fluorescent phospholipid labels are introduced, ZMWs. This is one ZMWs. Like this, there are millions of ZMWs in a plate, in a flow cell. So base being incorporated held for a million, tens of milliseconds and produce a light and it is captured. Now this is happening almost at the rate of 250 to 500 bases per second. And the phosphate chain is cleaved, releasing the dye molecule, and the whole process is repeated. This is a smart chip. Now this is how big it is. As small as you can place it on a fingertip. So there are thousands of ZMWs having a diameter of around 70 nanometer and depth will be around 100 nanometer. So width is around 70, depth is around 100 nanometer, and detection volume will be around 20 zeptoliters, 2 into 10 to the power of minus 21 liters. And this is a typical flow cell. They are multiplexed. There are 4 here, and each one will be like this. This is the scale of 43.5 micrometer, and each one is a ZMW. You can see how many will be there. So there will be millions of these cells in a chip, only this big. And they'll be doing parallel sequences. This is a typical chip. So this is a typical cell. Each ZMW, you have the polymerase attached there, and the long chain DNA is attached to that, and it starts sequencing. Single cell, this is going in, and the double stranded goes out. And as and when this is attached, it gets detected only around 39. So, yeah, this is the technology. So this is on the cell. On the bottom, we have the optics, which will give out the light source, emission, basic fluorescence, and it will measure what light it is coming out, and it will process. And then you have the big data analysis by bioinformatics, and it will figure out which base is after what base in which cell. So each of these millions of cells, they are captured, analyzed almost at the same time. So that is the PacBio system. So the two important systems, this is SQL and SQL 2. There are around 1 billion ZMWs in a chip there. Then the higher version, you have 8 million. And you still have more versions coming up with a more increased number of ZMW cells, SMRT cells. This is a typical flow from the DNA. You just fragment it, then repair the ends, you ligate the adapters, purify the template, anneal the primers, and attach it, bind to the sequence. See, there is no PCR amplification of the DNA. You need very small amount of DNA, and that is utilized. There is no amplification step here. This length will be very large, and it can go up to around 50 to more than 100 kilobases in length. So the advantages of this and the limitations: long read length that will allow for greater certainty in the overlapping, better resolution of repetitive ends. This can also sequence DNA with unusually high GC content, which was a problem in the earlier sequencing technologies, especially short read ones. But the main limitations for the PacBio where the lower throughput error rate was quite high. When they started, it was almost 13%, and the cost per base was also much higher, more than 10 times costlier than Illumina. But now, upon the release of the more advanced platforms with higher number of ZMWs, the sequencing chemistry and the advanced sequencing chemistry, the error rate has now come as low as 3%, which is much higher, still higher than the Illumina or Torrent. Illumina and Torrent, they claim 99% accuracy. So error rate is less than 1%. Here it has come down to 3%. Okay, the second one is what is called as the nanopore sequence, developed by Oxford Nanopore Technologies. So what this technology does is, you are threading the DNA through a microscopic pore in a membrane. So you are simply pushing the DNA through a pore, and what is coming out, that is measured by technology, by measuring the current which flows. So they determine the sequence of the DNA fragment by passing the DNA through a pore protein in a membrane. And they became one of the first companies to bring about portable sequencing machines. So they started commercially selling these products in 2015. Okay, and they are very small, which you can hold it on the palm, mini ion technology. So this is the basic principle. So you have a protein nanopore set in an electrically resistant membrane bilayer, and there is a current flowing through the nanopore by setting a voltage across this membrane. So now a standard current will flow. When a particular ion, if an analyte passes through the pore or near its aperture here, this will disrupt the current. The type of disruption depends on the what molecule which is coming. So this system can be used to distinguish the 4 standard bases, GATC, and also the modified bases. So when one by one the bases are coming out, this can detect the disruption in the current, can detect what kind of base is the one which is passing through. That is the basis of the nanopore sensing. So there is a porous transmembrane cellular protein, diameter of around 1 nanometer. This is actually a modified alpha hemolysin from Mycobacterium segmentis porine. That's the porine from this organism is the channel. Just got a transmembrane channel there, and you are passing the DNA. So double stranded DNA is stripped, and you allow one DNA to pass. So nanopore is immersed in a conducting fluid, and the voltage is applied across that, and you allow the DNA to pass. So when the DNA is passing, you see here the different bases, they disrupt the current to different extent, and based on that you do what is called base calling. If this is the one, then it is A. If this only this much is disrupted, it is C. This is T, like that. So depending on the disruption, it can identify what base is which is coming out. So continuously bases are coming out, passing through that, and that is getting. Now this is happening, remember, at the rate of around 250 to 300 bases per second. Very rapid base calling. So you have a library preparation. This is the workflow. Prepare the ends, add the ligate the adapters, size select, and then you load it into a flow cell. And this is the flow cell. Each one, this is the top view. Each one will contain a particular piece of DNA, and each one is a pore, and it is measured. So like that, this is a single nanopore cross section, and the DNA will be threaded through that, and it is read. So there are various types of machines they have come up with. Flongle, then Smith-Dion, which can be actually connected to, they claim to be connected to a smartphone. So using your smartphone, you can do DNA sequencing. Then you have the mini ion or grid ion and prometheon, which are the large scale ones. These are the smaller ones. So advantages and limitations of this. This is a mini ion, highly portable, and it is capable of sequencing when you can plug it in a laptop. On your small laptop, you can connect it and do the sequencing. And this is very particularly advantageous in the field. And when there was an Ebola outbreak in 2015, this was used in the field to sequence the virus strains. And this was also used to conduct first sequencing experiments in space, in ISS stations. And this is a label-free sequence determination, there is no label used here. Unlike in the case of SMRT, there is a fluorescent dye to be used. Here, there is no label. We simply pass the DNA through a pore and then read. So you can determine the native DNA and RNA sequencing without amplifications, capable of producing extremely long length reads. The limitations, again, there are relatively high error rate than the short read sequencing, almost close to 15%. But there is a lot of research and development going on in this field to improve the structure of the nanopore and the base calling, and they are reducing the error rate to much lower than what is there. So this is the comparison between the third generation platforms, PacBio and the Oxford Nanopore systems. They are the different machines like that. And this is the average baseline, the read length is actually 10 to 15 kilobases compared to NGS, which is only around 500 bases. And the error rate is 10 to 15%, and the runtime and maximum throughput up to 500 GB can be processed in a single reaction. And the Oxford Nanopore, we have different up to 4 megabases, 4 MB size can be read, and within a minute to within up to 72 hours, and 14 TB terabyte base size of a large amount of DNA can be read through this. So the major applications of the third generation and the what type of sequencing platform for the research interest. This slide indicates what can be used, which technology to be used for what kind of applications. For example, when you are going for whole genome sequencing for the de novo genome assembly, you can go for either PacBio or Oxford Nano. When there is mutation analysis, better go for NGS, next generation. Now, that is because mutation analysis, you are looking for specific mutations, specific bases. So wherever there is a need for very accurate base calling, NGS is still the preferred because error rate is very low. The error rate in these ones are relatively higher. So wherever you want to detect SNPs, small indels and all, it's all always the NGS. Construction of gene panels for SNPs, small indels, we always go for next generation sequencing where the error rate is very low. Otherwise, for targeted sequencing, whole transcriptome sequencing can use both PacBio. For the direct RNA sequencing, it is currently only the ONT because you don't use any enzymes, polymerase, or the label. So directly you pass the molecule, DNA or RNA, through the pore and then they detect. So Oxford Nanopore is the preferred technology. So due to the advent of all these third generation sequencing, the cost, which is now for the sequencing the human genome, has come down drastically. In 2001, it was more than 100 millions when we started. This was closed 2002. We got the first draft, so it was costing almost $1 million, 100 million for sequencing a human genome. Now it has come down drastically to less than 1 billion. No, sorry, $100,000. So it is coming down. The cost per raw megabase has also come down. This is the one. So it has come down to less than 1,000. So almost within $1,000, you can get your genome sequence. And there are now technologies, especially the Oxford Nanopore, they are going big into the diagnostics. Whereas PacBio is more into research kind of sequencing. Oxford Nanopore technology is going for the diagnostic ones, and within $1,000, you can get the whole genome sequenced for detection of any diseases and those kind of things. So what are the other methods which are under development currently emerging? So we have advances in the nanopore technology, solid state nanopores engineered from synthetic materials such as graphene or carbon nanotubes. Then they are converting this smidge ion, making it even more portable so that you can attach it to a smartphone. This is still in R&D, but it might come out soon in the market also. Then the single cell DNA. So taking the DNA from a single cell, isolating a cell and measuring, taking its DNA and sequencing from individual cells. 6 to 7 picogram of DNA is sufficient that can be measured. In situ nucleic acid sequencing, performing the sequencing within the intact tissues, that's also under progress R&D. Then microscopy based sequencing using the scanning electron tunneling microscopy, all this. So these are the other technologies which are coming up, which we sometimes call as fourth generation, more advanced than the TGS sequencing. So to conclude, I could say that even though this third generation sequencing technology, they came up almost 10, 15 years. No, not even 15 years. They started commercially in around 2015, so around 8 to 9 years. They have already established large-scale DNA sequencing and RNA sequencing applications, and which have taken the sequencing to a much higher level. So this the advancement may terminate the NGS sequencing era. Now, currently, the Illumina and the Ion Torrent, they are the major sequencing platforms. So with the advent of this third generation sequencing technology, maybe they might come down with more accurate variant calling and efficient identification of mutations. So although this third generation platform provide increased throughput and through analysis, the main problem is the data set analysis, big data analysis. That's a major challenge. And with the production of high accurate reads and introduction of more advanced bioinformatic tools, the TGS can evolve rapidly, and it can establish as a future novel platforms. The enhanced version of TGS or the brand new fourth generation technology with an unprecedented characteristics can also come up in future. Yeah, so that's it. So currently we have all different technologies. These users who can choose what kind of technology platform they want, and then do the sequencing. Any questions?
Hello, sir.
Yeah.
So, thank you, sir, for valuable information. Sir, myself, Kavita JC, Davangere University. Sir, my question is, when you compare pyro sequencing with